Okay. I want to review an action potential. We're going to start from the beginning, walk our way through it. This is how you guys need to start studying for the class. I know we go through a lot of different material, but what it's doing is giving you building blocks for the bigger picture. I can't just throw out action potential and say, this is what happens. You have to know how voltage-gated channels work. You have to know how resting memory potential is calculated. What's the most important ion that's there? It all goes into this one picture. So what we'll do is we're going to go through it, we'll review it step by step, and just shout out again what's going on, what's happening, and we'll go through the entire process, and it'll make sense. So we're talking about action potentials. Mainly nerve and muscle. Heart, cardiac action potentials are a little bit different. We're going to discuss that more when we get to cardiac. But all nerve and muscles will have the similar action potential. So on the y-axis, we have our millivolts. Okay, so we have -70, -55, 0, and +30 for our lines. -70, that's our resting membrane potential, right? That's where we are at rest. So nothing is happening, and we're good. This is rest. What ion is responsible for that -70? What's the biggest one responsible for that? Potassium. So potassium is why we are at -70. -55, what does that represent? That's our threshold. Once you hit threshold, it's all or none, everything happens. So threshold, we have that line. Okay? So now we start moving up towards threshold. So we're starting to creep up here. Why are we starting to creep up and get more positive? What is going on? Sodium is moving, right? So in order for sodium to move, what has to happen? The gate has to open. When do sodium channels open up? When do, like what millivolt? Around -70, -65 is when those sodium channels start to open. Once you get enough sodium channels open, we start to depolarize, right? So as we depolarize and we hit threshold, we go straight up. Rapid depolarization. Why is it rapid? What's happening once we hit threshold? All the sodium channels open up. So only sodium is moving, right? What about potassium? Is potassium moving right now? No. Why is potassium not moving? Gate's closed. Nothing's going to happen. So only sodium is moving, and this is our depolarization. Okay? We start creeping up. We get above. I'm going to get rid of these words here. Hang on a second. Okay. Okay. We come up, a little bit of an overshoot, and then we start to come back down. Okay. This is all dipole. We get to the top. What happens as we get to the top now? Now we are above 0 millivolts. We're in the positive range now. What happens to the sodium gate at this point? Does it close? The inactivation gate pops into place. The gate is still open, but the little inactivation gate gets in the way, stopping sodium from moving. So there's no more sodium happening now because the inactivation gate popped in. But we're at positive millivolts. What gate opens at this voltage now? Potassium starts to open up. So which way is potassium going to move? Out of the cell. It's going to leave the cell. So as all these positive ions leave the cell, we start to repolarize. Repol. And what is responsible for repolarization? Potassium. Okay, so now we are repolarizing the cell, going back down to resting memory potential. But when potassium moves all by itself, it's going to want to go to like negative 90. It's going to keep dropping. And eventually, when we get down below negative 70 again, what happens to the potassium channels at this point? They close. Potassium channels do close. There's no inactivation gate on a potassium channel, just the sodium ones. So I'll put sodium on that side. We have potassium on the other side. So we're going to bring it all the way back down. We do have a little bit of a hyperpolarization in there. But it's going to come back up to resting memory potential. That's all the action potential is. But you understand how you had to understand how gates work. You had to understand concentration differences. You had to understand electrical chemical flow. All those are pieces to understand this picture right here. So everything we did in general phys, basically that whole section, was being able to describe this picture. Hopefully that was a good review from general phys regarding action potentials. Okay. Now the last thing I do want to mention with action potentials, and let me erase this part. One thing we haven't talked about, again in much detail, are the refractory periods. So I do want to highlight the refractory periods on this graph. We have two types of refractory periods. What are they? Relative and absolute. The absolute refractory period, nothing is going to happen. No matter how big of a stimulus you give, if you are in the absolute refractory period, nothing is going to happen. Why will nothing happen during an absolute refractory period? What is special about that sodium gate? It has that inactivation gate in there. You have to wait for the sodium channel to reset before you can have another action potential. The inactivation gate is going to be in the way. We have to come back down to like those negative millivolts for everything to reset. So, let me put it in green. Hope it stands out. This box, which covers everything above threshold, oops, it all got taken away. Okay, this whole box, and let me just redraw that. Okay. That whole box is the absolute refractory period. We have to wait for that line to get below threshold again, then the sodium channels start to reset back to normal position. So that is the absolute refractory period. We also have the relative. The relative refractory period starts once the absolute is over, and it usually goes until you hit that hyperpolarization mark. At this time, enough sodium channels have reset where you can have another action potential occur. So that's the relative refractory period. Okay. Some sodium channels reset when it gets below threshold again. The sodium channel has to go back to normal setting. When enough of those have gone back to normal setting, the whole process can start over again, and if a big enough stimulus comes in, you'll have another action potential. So those are the big steps that you have to know as far as regarding action potential. Know absolute, relative refractory periods, know the big differences why. Absolute, none of the sodium channels have reset. In the relative period, some, because they all don't open up at the same time, each of them have different properties. Some will open up sooner than others, and it'll gradually bring you up. But once you hit threshold, then it all starts. Everyone good on this one? Any questions? Potentials. Okay. So we talked yesterday or last week about sensory with regards to touch. So make sure you know what all those different receptors and what they transduce. So Merkel's discs, Pacinian corpuscles, free nerve endings, all the little things in our sensory that allow us to sense the outside environment. Make sure you know the definitions. Now we're going to go through how we see and what goes on. So the big transducing cells are the photoreceptor cells. Photoreceptors takes photo light and converts it into an electrical signal so that our brain can decode what we're looking at. Visual field is very highly ordered, so PAs, you'll start tomorrow, you're going to go over your ENT exams. You did ophtho last week, right? Okay, so we're going to go through a lot of things that you guys will be checking on your exam. So it goes to the retina, and then we'll see what happens. How does the light, or the impulse, the electrical charge, get from the retina to our visual cortex so we can actually interpret what we're seeing? So the central processing occurs in the primary visual cortex, which is in the occipital lobe, which is in the back of the brain. So when you are looking at things and you're reading whatever you're reading, that image has to go from the back of the retina and go all the way to the back of the skull so that your brain can know what you're looking at. Which is why when people, like concussions in football, and they lay on the back of their head, they see stars and weird things for a while because you hit and you really bang that occipital cortex hard, which is where your visual cortex is. So it gets a little stunned and you get problems seeing. So the different qualities of vision, we can look at the form, the color, the depth, motion, and distance. So depth perception, how far away is something? Your eyes play a big role in that. So here's our anatomy of the eye. This yellow line shows how light goes into our eye. We have different things that we'll go and we learn about. Ciliary body we're going to focus on. The aqueous humor is just the liquid inside the eye, keeping the eyeball puffed out and solid. So that jelly inside. We have the lens, the iris, which controls how much light gets into the eye. And then we have at the back here, we have our retina. And that is actually what picks up all the light. So the lens helps focus it onto the retina so that you can have a good image so you know what you're looking at. If you have blurry vision, it's very hard to see, they get very cloudy, that's a lens issue. If you're looking at things and it feels like pixels are gone, like you look at a TV and you have dead pixels, that means you have a retina problem because the retina is what picks up the image itself. So if it's blurry, it's a lens problem. If you're missing images and it's black in certain pixels, then you have a retina problem. So fluid compartments of the eye, we talked about CSF. Similar process here. We're constantly making aqueous humor and we're constantly reabsorbing it. And as long as the production of it matches the removal, then your pressure in your eye is normal. Okay? For example, glaucoma is increased intraocular pressure because you have too much fluid in the eye and the pressure goes up. So that's what glaucoma is. It's too much aqueous humor increasing the pressure in the eye. We talked about that already. As far as secretion and absorption of the humor, the epithelial cells is what actually creates the aqueous humor and secretes it into the eye in the posterior compartment, flows through the pupil into the anterior compartment, and then it leaves through the canal of Schlemm. So the canal of Schlemm in the eye is what drains the aqueous humor. So we have constant flow. Okay? And it drains into the extraocular veins. So that's how we reabsorb aqueous humor, and we create it all the time. And here's our glaucoma. I talked about that already for your correlation. When you look at an image, Because you are going through a lens and you're going through water, essentially, or a liquid, the image on the back of the retina is flipped and turned upside down when you actually look at things. Your brain takes it and converts it back to a normal upright image. That's why I said also last week about babies having to look at mobiles and playing peekaboo so they know what's up and down with regards to vision. So there is some refraction going on. Because we have a curved surface, we're going through liquid, we get some refraction. So we have this thing called refraction of light, and we have our focal point. And if all the light sources are able to converge on one point in the correct location of the retina, then you have good vision. If you can't see far or you can't see near, this focal point is in the wrong place. And we'll get to that in a second about what happens. The distance beyond the lens where light focuses is the focal distance. So again, the projected image is turned upside down with the two sides reversed, but our brain turns it back to normal so we know we are upright and whatnot. When you guys start using that diopter to check in people's eyes, there's a setting that you do on your end, and what you're doing is you're correcting your vision problems with that diopter. So when you spin that before you go into someone's eye, you're correcting for your bad vision because you can't use glasses with that ophthalmoscope. So it corrects your vision so that you can see clearly in their eye. Accommodation. You guys did this already, right? Okay. What is accommodation? What is it? You got to know on Wednesday, right? Okay. We're focusing on an object far away or close up. Okay? That's what accommodation is. So what you're supposed to do is have them stare at your pen, and then in the same line, tell them to look over your right shoulder. Okay? The goal for that test, you don't want their eyes to have to move to a different location. So I know you're right in front of me, so and people in this row at least. If I put the pen over my right shoulder, say focus on the pen, now look over my shoulder, your eyes don't have to move very much. So when you're doing this on your exam for accommodation, just don't say like, look at my pen, now look over there. They're looking over. You want to look at their pupils and see how their pupils change when they look at near and look at far. A lot of you right now are probably reading a lot more on your screens than you probably had in the past because a lot of the material you got to learn. Accommodation really is a problem with near vision. It causes a lot of eye strain. So there's a rule, it's called the 20-20 rule. For every 20 minutes that you're looking at your computer screen, you want to look for like 20 seconds at something 20 feet away and let your eyes relax. If you do that, you'll feel your eyes like, okay, I can feel a little tension going away. But that's where a lot of strain and like ocular strain comes in because you're so focused on everything right in front of you that you're accommodating so much, it causes eye strain. So what are we actually talking about? It's this muscle right there, that ciliary muscle. Okay? Contraction of the ciliary muscle reduces the tension and allow the lens to assume a more rounded shape. This is controlled via parasympathetic nerves. Okay, so we're going to start going through, especially with the eye, parasympathetic innervation and sympathetic innervation. What's the difference? What's going to happen to their eye based off of what nerve is causing the issue? So this is our ciliary muscle, and it's involved with accommodation. So when you look at long, far distances, kind of tightens up a little bit. Looking at near distances, ciliary muscle relaxes, reduces the tension, but it also causes a little bit of tense in the muscle surrounding the eye, and that can cause some eye strain. So make sure you understand ciliary muscle is used in accommodation. When the ciliary muscle relaxes, it allows near vision. When the ciliary muscle contracts, it allows far. Errors of refraction. So how do we see things? The word emetropia means normal vision. if you have what's called myopia, you're nearsighted. Hyperopia is farsightedness. We have presbyopia, a reduced ability to accommodate near or distant objects. So when you do your accommodation and you look at their pupils, you're not going to see them change very much with presbyopia. And we have astigmatism, which is an incorrect curvature of the eye in one place. So if you have an astigmatism, things are wavy, and I think I have a picture. Yeah, the picture there. So this was the normal one. The light comes in and it focuses right there on the retina. This is what happens in normal vision. With myopia, nearsightedness, that is where the focal point is. You see how far into the eye it is? This is nearsightedness. Okay, and then you correct with glasses, and now the focal point is back where it's supposed to be on the back of the retina. So, correction of lenses. When you are doing your eye exam, you have to note, do they wear lenses or not when you did it? Do they wear corrective lenses? Do they wear glasses or contacts? Just documentation that, yes, I know they have corrective lenses, and with their corrective lenses, their vision is fine. So that's myopia. Here is your hyperopia, your farsightedness. Look at where that focal point is. It's beyond the retina. So the image that's being projected onto the retina is not focused. You're going to have vision problems. So this is what hyperopia is, or farsightedness. You give glasses, you correct it, and now the focal point is back on the back of the retina where it's supposed to be. And that's how corrective lenses work. Contacts do the same thing. So those are the different types of vision problems we have. Here is what astigmatism looks like. Okay, it can be vertical lines are blurred, horizontal lines are blurred. But basically there's a blurriness or a waviness to some of the lines. There's like a chart that has like a grid pattern on it. You can look at that and you'll see the grid. The lines will be wavy. So that's astigmatism. Okay, moving on to the retina. So how do we actually see and process our imaging, the colors, depth, all that stuff? So we have this thing called retinal pigment epithelium, and that's the outermost layer of the retina, and this is where they have the phototransduction occur because we have rhodopsin here. Rhodopsin is a chemical that's in the retina. And when vision and light hits the rhodopsin, it changes. And we're going to go through this and see what happens as we go through how the light is processed into electrical signals and what happens. We then have retinal bipolar cells. They form a synapse with the photoreceptors and the ganglion cells. This is one of the only cases where we have this bipolar neuron, which looks like this. And there's a cell body in the middle. So it's getting information from bipolar cells and, well, it should be the ganglion. Ganglion cells and then retinal bipolar cell, which is really just the photoreceptors. So we're getting information coming from two different ways. The ganglion cells is the output cell of the retina, and the axons all collect to turn into the optic nerve. So the optic nerve starts off as all around the retina, and then all those ganglion come together to make an optic nerve. That's what actually comes out of the eye. We have horizontal cells, allow for radial orientation to form synapses, and we have amacrine cells, and they're in the inner layer of the retina, and I have pictures that can describe all this as well, so we'll go through it again. So here it is. Light hits this part. This is what you guys see when you do your fundoscopic exam. How many of you actually saw the lines, the veins? No one does. I know no one does. It's okay. It's very, very hard to see. You need a really dark room. You need those pupils to be really dilated. And if you go to the ophthalmologist, they cheat because they give you dilator drops. So they can really open up that pupil wide so they can see everything inside. So light hits that retinal layer. Then We have our ganglion cell there, which then connects to our bipolar cell. And it's all blown up right here. Okay, so here's our pigment epithelium. These are the rods that contain the rhodopsin in it. There's our bipolar cell, which takes information from the pigment epithelium and sends it to the ganglion cell. And all these ganglion cells that you see right here in the blue, they all connect to make your optic nerve. So all these ganglion cells all come together to create the optic nerve, which then goes to the brain and then follows the whole pathway, which we'll get to in a second. So this is the order of what happens. Pigment epithelium changes, make changes in the rods or the horizontal cell, which then goes to the bipolar cell, ganglion cells, optic nerve, and we'll go from there as we go. Properties of rods and cones. So this is how we actually see color, black and white, low light, how we see better in low light, all has to do with these rods and cones. Rods, monochromatic, highly sensitive to light, and rods allow you to see well in dark light. So we have a dark adaptation, increased light sensitivity after prolonged darkness, and that occurs in the rods. So if you've been in a dark room for a long time and you go to a bright light, it hurts and your eyes go crazy because we've adapted to low light. Anyone know why pirates wear patches?
Easier switching.
So what they do is they wear a patch on their right eye when they go down below in the decks because it's dark. And when they go back out to the light, they switch where the patch was. So they avoid this whole accommodation thing. Because you know when you go to sleep at night, it takes like 15 minutes, 10 minutes before you actually see well in the dark. There's an adaptation process there. You don't want to have to keep doing that, so they switch the patch. It allows them to see better at day and then dark time. Cones. Cones are the color. Less sensitive to light, but they have light intensity. So depending on what strengths of different cones are being stimulated, gives you different sensations of colors. Blue, or the S cones, are short wavelength, green are medium, and red are the long. So if you have very high red, low blue, low green, you'll see more red. If you see a lot of red, a lot of blue mixed together with a little bit of green, you'll get more of a purple color. It's all just like your primary colors. You mix them up in different strengths and you get different colors. Disney messes with your color perception. Okay? When you go to Disney, what color is the ground? Red. It saturates your red receptors, so when you look at everything else, everything looks really green. It's done on purpose. It's to trick you to make sure the grass looks so much greener because you're saturating your red cones. So everything looks green. They also do it with the color of their buildings. The color of their buildings are very muted, so they blend in with the sky, so you don't even notice the buildings are there. So they play with your rods and your cones to screw around with your perception of color at Disney. So again, the different perceptions, the different strengths, just understand what rods do, know what cones do. You don't have to know what wavelength the blue one goes on. That's not important. Just put it there. People like to know these things, but I'm not going to test you which one is that. Know what the rods do, know what the cones do. So dark and light adaptation. How well do we see in the dark? We have to change the sensitivity of our retina to the changing light sources. In bright light, rhodopsin dissociates, so we have less visual pigment available. Okay, so bright light, rhodopsin dissociates and it goes away. Less pigment available. In dark light, the rhodopsin stores are built up, more rhodopsin, more phototransduction. Remember, we have to convert a visual stimulus into an electrical one. With the light into electrical. We need phototransduction. These are the cells that help with it. So rods are responsible for dark adaptation. So if someone has color blindness, where's their problem? The rods or the cones? Cones. If they have problem with dark light, dark rooms, they can't see very well, they don't adjust very well. Where's the problem? Rods. Very good. Vitamin A helps maintenance of healthy epithelial and vision. That's why I always say like, carrots help with your eyes and whatnot. But it helps with night blindness and stuff like that. You know, again, it's not in your test. It's just reinforcing certain things. So color vision, we decode 3 different types of colors. And if more green cones are stimulated, then you get green. Again, it's all, like if you ever like get paint mixed at a store, they put, you want a color of this color, but they put like different color in different quantities into the paint before they mix it. The same thing with your rods and with your cones. Different colors are saturated more, gives you different color that you see. So here's an example of different types of color blindness and what they see. So in protanopia, they're red, green, weak. So you can see they don't see very much red or green. Again, just showing you different color blindness. It's not going to be on your test. The visual pathway. When you are studying this section, just pretend you're a photon of light. How do you go from what I'm seeing outside? How does that light get into the eye? And then from the eye, where does it go from there? How do I get to my primary visual cortex? And that's what we're going to learn right now, how we get there. So the light comes through the cornea, anterior chamber, anterior cavity, through the pupil, through the posterior chamber, and then finally we get to the lens. So we haven't gotten to the lens yet. Now we're at the lens. So the lens, the light will go through the posterior cavity, and then finally will hit your retina. Once we are at the retina, we're going to activate those pigmented cells. Going to trigger those rods and cones, go to the horizontal cells, the bipolar cells, the amacrine, the ganglion cells, and eventually that ganglia all converge into one big nerve called the optic nerve, which goes to the brain. We're going to take over from that optic nerve, and where does it go? So main visual pathway, axons from the ganglial cells enter the optic nerve in each eye. I'm just describing what I'm going to be talking about. We're going to draw out in a second. Optic nerves meet at that optic chiasm, where some axons will cross the midline. So the optic chiasm. Anyone know what, I'll put tissue or, not an organ, but what's in that optic chiasm? Anyone know what tissue, I can't remember the word right now, a structure is in the optic chiasm? Go to your anatomy, and if you don't get there yet, you will. In the optic chiasm lives the pituitary gland. Sits right on top. So if you have a pituitary tumor, that tumor gets big, and guess what it does? Compresses that optic chiasm, and you get different visual problems when you have a pituitary tumor because of this problem right here. The optic tract leaves this optic chiasm into the lateral geniculate nucleus of the thalamus. Remember we said last week, all sensory has to go through the thalamus. Vision is a sensory, it's got to go through the thalamus, and the part of the thalamus that it goes through is called the lateral geniculate body, or lateral geniculate nucleus. All the same thing. It means that's the part of the thalamus that the vision travels through. There are different parts of the thalamus that other sensories go through, but vision is the lateral geniculate nucleus or body. Then, once we leave the thalamus, we have second order sensory that go to the primary visual cortex via optic radiations. So we have a lot of nerves that branch out, and they all innervate parts of the primary visual cortex so that we can interpret our vision. We do have overlap from right eye and left eye. There is some overlap in our vision to produce binocular vision. Here's a real good example of that right now. When both eyes are open, do you see your nose? Close one eye. The nose comes right back. We block it out because we have dual sensory. If you got rid of one eye, the nose comes back because there's no more depth perception between the two eyes. And the nose comes back into frame. So here are the eyes. There's our left. There is our right. I'm going to put a line going across. And we're going to break this right down the middle. So we're going to define the very top here. And this is where your anatomical landmarks and definitions come into play. We're going to talk about it right here. We have lateral vision, medial, and then we have medial on the right, and we have lateral vision on the right. I choose red and blue, so two different colors. So we have our vision, left lateral, medial views right in the middle, and our lateral views and how they come into our eye. So if I say our right lateral vision projects onto what part of the eye? The nasal side. So this is going to come in here. We have nasal, lateral, nasal, lateral. So the right lateral vision, when it comes into the eye, it goes to the opposite side. So left lateral is going to go to nasal or medial, whatever you want to call it. Medial, medial. Okay. Conversely, same thing happens on the opposite side. Left lateral vision comes to the medial portion of the retina. So this whole section here is just retina. The top part is our vision. Okay. Conversely, the medial vision is going to project onto the lateral retina. And our right medial, so right where I'm my center vision, is going to project onto the lateral retina. Everyone good so far? Understand how everything flips onto the retina wall. The reason why my vision is so bad is because I had a retinal detachment in end of March. So the retina came off the eye, so I couldn't see half my vision because it was gone. So they had to do a surgery to reattach the retina back to the eye. And when they do that, they suck out all the aqueous humor, right? They have to go and attach it. So what they do is because when you cut yourself, what do you do to stop the bleeding? Pressure, right? How do you put pressure on your eye? Can't. So they put a bubble there, and they inflate the bubble, and it allows the compression against the retina to push it against the wall. But over time, as our aqueous humor, we create that stuff all the time. Make sure you know what cells create your aqueous humor. As that cell fills in, the bubble gets smaller and smaller and smaller. So, from where I am right now, where do you think the bubble is hanging out in my eye? Anterior or superior and inferior part of my eye. If it's a bubble, it floats, right? So the bubble is going to be the top part of my eye. Where do I actually see it in my vision? Bottom, the inferior part of my eye. So I have a little bubble that's floating around the bottom of my vision, but it's really at the top of my vision because everything's flipped, right? So that's where I see the bubble. That's why I'm like, I can't see very well because my vision is bad and I can't really see far. So I can't see close either, but figure it out. Siri's my guide when I drive. I'm like, turn left, I turn left. That's how I figure it. It's not that bad. Okay. Now, okay, I don't want to mess this up. The nasal, the lateral, okay, so we're here. The lateral vision on the right projects on the nasal or medial portion of the retina. Then, this nerve, remember this is the optic nerve, comes down and crosses the optic chiasm to go to the thalamus. The converse happens on the other side. The lateral vision comes in and it crosses the optic chiasm and it goes into the thalamus. What is the specific location in the thalamus, the collection of cells that process our visual? Yep, lateral geniculate nucleus. That's in the thalamus. Now, the medial vision stays on the side and it goes to the thalamus. The medial vision projects on the lateral retina, stays on that side. So, what vision is going and crossing in the middle? What kind of vision? Lateral vision or medial vision is going through that optic chiasm? Lateral. Only the laterals, right? So, if I knock out that optic chiasm, and I draw some eyes here, what kind of vision am I going to lose in my left eye? Lateral, right? What's going to happen to my right eye? What kind of vision am I going to lose? Lateral. All I can see is my medial, which is called tunnel vision. Because the optic chiasm is what allows those pathways to cross. If I block it, I'm not going to be able to see that side of my vision. Now, once you are here, these are the optic radiations that many different nerves, and these all go to the primary visual cortex in the occipital lobe of the brain. This is the pathway that light takes. So, when you're studying this, just pretend you are a photon of light. I am a lateral vision on my right. It's going to project on the medial surface of the retina, which is then going to cross the optic chiasm to go to the opposite side thalamus. And then from there, the optic radiations, I'll put here. Optic radiations or whatever, there's a lot of different nerves, all go to the occipital area to allow your brain to decode what you're seeing. So the colors are there for a reason. Make sure you follow the same color, or we don't care what color you use, but make sure you follow that the same colors follow the same thing. Okay? Any questions about the vision pathways and what direction the light takes? Okay. Eye movements, how do we move our eye? Just remember this there at the bottom. I'm not going to ask you anything else. LR6, lateral rectus, cranial nerve 6. SO4 is superior oblique from the 4th cranial nerve. And the rest are 3, the oculomotor. So I'm not going to ask you what direction they go in. Just know LR6, SO4, R3 with regards to eye movements. So when you guys are doing your EOMs, right, you hold the pen. Follow the pen with my light. I am looking to make sure all of these muscles are working. And if the eye does not do certain activities, then you can figure out what muscle is weak that does not allow the the eye to go all the way to the right. That's have different cranial nerve palsies. People, they're looking at you, but the eye is looking the other way. That's a cranial nerve palsy. The muscle's not working, therefore nothing's able to abduct the eye or adduct the eye. Okay, so for right now for this class, just know those things. That's all I gotta know. Autonomic innervation of the eye. So let's take a break. Come back at 1:35. Okay, let's get started. We're gonna finish. We'll finish nerves today, neurophysiology today. On Thursday, we have a quick lecture on immunology, and then we'll do a review for the exam on Thursday. On Sunday night, I'll do that live session review. Just ask questions. I'll draw it out like we do here. Any concepts you're stuck with, I'll go over it. I gave you all my contact info, my email, and my room number. So if you have any questions, email me, come see me. But obviously, you guys are all invited as well on Sunday night. The exam on Monday, 50 questions, multiple choice. There's no trick questions. All the information you need is in the question. You don't have to think, well, is he talking about this? No, I'm not. If it's in the question, it's in the question. If it's not, it doesn't exist. So it has to be there. They're going to be straightforward questions. Remember, I told you this semester is all definitions. Know the language of physiology, language of medicine. So I'll say like an example of a question. During depolarization, which ion is moving? Sodium. That's the question right there. That's all it is. Or I may say during what ion is moving during repolarization? Potassium. Okay? Those are the questions. And then I'll add some thinking questions based off of concepts that we learned just to get your brain working, how you should start thinking about issues. Okay? So that's the exam. Now, autonomic innervation of the eye. Okay, we talked about autonomic nervous system. What are the two parts of our autonomic nervous system? Well, there's really three, but what are the two? Sympathetic, parasympathetic, and that gut braid of the enteric nervous system, which we'll talk more about when we get to GI. Okay, so we have brain sends these signals to the oculomotor nucleus in the midbrain. Oculomotor is nothing, the cranial nerve. Which nerve is that? Three. Okay. We have the motor nucleus, gives rise to motor fibers in our EOMs. And we have the visceral part, which is the parasympathetic preganglionic fibers. And they go from cranial nerve 3 to the ciliary ganglia. And we'll go through each part in a second here. So, here is your pupillary reflex. So you shine a light in the eyes, and you look at one eye, you shine the light in the eye, you look at the other eye, and so on and so forth. What are we doing when we do that? So cranial nerve 3 is our oculomotor nerve, helps control our pupillary reflex. With meiosis or constriction, shining bright light, what should happen to the pupil? It's going to constrict. It's too much light. The pupil is like the shutter on a camera. How much light do you let get in to the camera? So during meiosis constriction, it's a parasympathetic reflex. So look right here. Parasympathetic stimulation causes the circular muscles to contract in the eye, in the pupil. The circular muscles. And we get pinpoint pupils. So, this is parasympathetic. What neurotransmitter goes along with parasympathetic? It's also known as a cholinergic neurons. Acetylcholine is the neurotransmitter. So if someone's taking a lot of acetylcholine, okay, a lot of parasympathetic innervation, what's going to happen to their pupils on exam? They'll be constricted. Okay? So when you're looking for constriction of the pupils, it's a parasympathetic response. Now, mydriasis, or dilation of the pupil, getting very, very wide now in dim light. So right now, people in the back corner, dim light, the pupils are going to be big. If you're closer to the TVs or the light, your pupils will be a little bit smaller to allow for a certain amount of light to get in. Dilation of the eye is a sympathetic nervous system response. And when you have sympathetic stimulation, it causes the radial muscles to contract. So you get huge dilated eyes. Okay. Anyone coming in with like cocaine overdose? Cocaine is a sympathomimetic. It mimics the sympathetic nervous system. So what do you think the pupils are look like with people coming in with cocaine overdose? Wide, huge eyes. Because you gave a sympathetic drug that's going to stimulate the sympathetic nervous system, and your pupils get huge. So those drops they give you in your eyes to dilate your eyes, what kind of nerve stimulator is it? Sympathetic nerve stimulator, because it's going to make your eyes wide. So you have to know that part for everything, obviously for your PD exam. And so now you understand what you're actually looking at when you shine light in the eye. You're testing for sympathetic and parasympathetic innervation. So to review, circular muscles cause constriction. Radial muscle, dilation. And that's how our eyes get bigger and smaller, through different autonomic innervation stimulation. Okay? And that's how our pupils react. Okay, that's really it on visual. Now we're going to move on to auditory and vestibular. So how do we hear and how do we balance ourselves? So hearing and balance, what cranial nerve deals with this one? 8. What is the? Vestibulocochlear. Vestibulo refers to what? Balance. Cochlear refers to the hearing. So they're both involved with balance and hearing. Also has some complex higher processing that allows verbal communication, so you can hear, you understand what I am saying to you, you comprehend what I'm saying to you, and then through higher processes you can respond to me in an appropriate way. So the vestibular system informs the nervous system about position of the head and how it's moving. So if you ever go on roller coasters and you get sick afterwards, you'll find out why in this lecture. We're going to go through it, why you feel like crap afterwards. We both, whether it's vestibular or cochlear, all use these hair cells to transduce the mechanical force into electrical one. Remember, for all of these sensory ones, you have to convert the mechanical force, so my voice goes into your ear, and it's moving your hair cells and letting you know that you're hearing something. Hair cells are bathed in a fluid-filled organ called the membranous labyrinth. And we have 2 labyrinths in each ear. We have the auditory part and we have the vestibular part. So those are the 2 different parts of our ear. And this is kind of like a drawing of what it looks like. We have our inner ear over here. We have the outer ear is there. The big difference between the outer ear and the inner ear is the tympanic membrane. That obviously seals it off there. So the eardrum or the tympanic membrane starts the difference between the outer ear and the inner ear. Here are, in the inner ear, we have the cochlear canals and we have these semicircular canals. And you can see how they're all in a certain way there. The cochlear portion, if you think about the sound coming in, the vibrations go around and around and around. And that's how we hear something, the vibrations that go through the whole system. These semicircular canals are set up in certain ways to allow directional movement. So I know if I'm walking forward, I know if I'm on my side, I know if I'm on this side, all because of these semicircular canals. They help with position sense. Where's your head? We also have, if you look here, we have our vestibular nerve, our auditory nerve, the whole thing together. The facial nerve is right here as well. Okay, so the facial nerve also travels through this area. There's a disease later on you'll learn about. It's called an acoustic neuroma. Basically, it's a tumor cell of the acoustic sheath, but the facial nerve is there as well. So if the auditory nerve gets bigger, what's it going to compress? Facial nerve. And if the facial nerve is messed up, they're going to have problems with their face. They may have pain, may not be able to use certain parts of their face because the muscle is down because the nerve is being compressed. And we have these little eustachian tubes. These eustachian tubes connect the ears to the nose and the eyes, and they help move stuff around. So if you ever get like sick and you feel fullness in your ear that you can't hear very well, it's because all the snot and everything that collects in the nose, everything, these eustachian tubes, they can build up, and the fluid will work back up into the ears, and you get an ear fullness. So that's where the eustachian tube comes in. Nature of sound. Sound travels in constant through the air. The way we can vary sound is through frequency modulation or amplitude modulation. Okay, on your car stereos in your car, you have a function called radio. No one ever uses it anymore, but it's there. You can look at it. And on the radio, you have two different settings. What are the two different settings on a car radio? AM and FM. Why do you think they're called AM and FM? FM is frequency modulation. AM is amplitude modulation. The reason why, if you ever drove in like different areas where they have a lot of mountains and hills, you don't get AM radio because it can't get through the mountains. And that's why you have different like FM, you get better coverage than AM. But just giving you things to why it makes sense. So that's how we can change our hearing. Our pitch to high or low tone we perceive, and the amplitude of the pressure reflects the intensity of the sound, and we perceive it as loudness. So amplitude modification gives us loudness. Frequency modification determines the pitch. Okay, so how high or how low it is based off the frequency. The loudness is the amplitude. Outer ear, sound waves are funneled, so the way our ears are shaped allows the sound to go through our pinna, the outer auricles, the helixes, into the external auditory canal, where it's going to start bouncing off that tympanic membrane. It's just a drum head, and it just bounces based off of the sound. The middle ear, between the tympanic membrane and the cochlea, has three bones or ossicles, the malleus, the incus, and the stapes. How many of you saw it on your physical exam? We haven't gone there yet, right? You'll do it tomorrow. Tomorrow's your ears, right? You did ears? Yeah. You didn't see the bones? Okay. As long as you saw one of them, you can see all of them. You might see some, not all the time, but you might see some bones. So, I'm talking to you, the sound is causing a vibration in the ear, and these vibrations are transmitted and amplified along those bones. Okay? The stapes is attached to the oval window. The big thing I want you to know is the purpose of the stapedius muscle. The stapedius muscle is like a control. If the sound gets too loud, the stapedius muscle can get activated and dampen the sound, the vibration, so the sound isn't so loud. So that's what the stapedius muscle is for. The inner ear. So now we're actually the part where the vibration, the hair cells are that allow the transduction of a mechanical sound to an electrical signal that our brain can now interpret. What I want you to know is just this bottom area down here. This structure, B, is called the organ of Corti. And this is the actual apparatus that allows you to hear things. Okay? We have this tectorial membrane. Here are the outer hair cells. And you can see here are the inner hair cells. So these hair cells are what move around and cause vibrations. As these hair cells are moved, you see the nerves there. There's the inner ear hair nerve. And these little things convert the mechanical force to an electrical signal that our brain can now interpret as sound. So the organ of Corti is the apparatus that converts the sound waves to an electrical signal so our brain can interpret what it is. We have these hair cells, and they start moving, and that allows you to know that you're hearing something. So again, organ of Corti has all the auditory receptors in it. We have these sensory hair cells, and they project into the endolymph of the cochlear duct. Endolymph is very important to remember, and we'll get to why it's important in a minute. So it converts sound waves into electrical impulses, and then the cochlear portion of the cranial nerve 8 transmit it to the brain, and you can hear things. So when sound waves enter the scala media, the tectorial membrane vibrates, which is what that part is right there. That tectorial membrane starts to vibrate, and it triggers those hair cells to start moving. And it bends what are called stereocilia. What are cilia? Little hairs. So we have little hairs. This is the outer hair cells and the inner hair cells. We call them stereocilia. Stereo sound allows us to hear. So we have fluids in our inner ear as well. We have what's called perilymph, which is similar to your extracellular fluid. Low potassium, high sodium. And we have the endolymph. And I said you got to know what this one is. This one is different than other parts of the body. Has very high potassium and low sodium. The difference in the potassium in the endolymph, in the ear, is what causes the depolarization to occur. So I said the majority of all the active potentials, sodium is what causes the depolarization. The only time it doesn't is in the ear, and in the endolymph it uses flow of potassium to cause the depolarization to occur. I don't know why in the ear it switched to potassium versus sodium, but it is. So that's the only time that a depolarization is caused by something other than sodium is in the endolymph. So if there's a question on your exam and I said, in the endolymph, which ion moves to cause depolarization? Answer, potassium. If I said, in an active potential, what causes the repolarization or depolarization to occur? I didn't mention anything about endolymph, sodium. Okay, I will mention that it's the endolymph that uses the potassium for depolarization. So, transduction of sound. Here are the cilia. Okay, the kinocilium or stereocilia, same thing. But these are the hair cells. As the vibrations occur and these hair cells start vibrating, that's the mechanical force. It converts it into an electrical signal by flux of potassium. Okay, high potassium here. As the hair cells start to move, it causes depolarization to occur due to potassium. As that depolarization occurs, we go down. Oh, there is our calcium voltage-gated channels right at the end. And what's that calcium going to do when it comes in the cell? Exocytosis, right? That's the whole purpose of calcium. Calcium comes in, we get exocytosis, now we've stimulated the cochlear nerve, and you're going to sense hearing because of those stereocillar bending based off of the mechanical forces of my voice talking to you guys. So that is basically what we talk about here. As the stereocilia bending, you bend the stereocilia towards the kinocilium. So there's the kino. As it goes towards it, you cause a depolarization. Okay? Everyone good so far on this part of hearing? Okay. We have inner hairs and we have outer hairs. Inner hair cells, transduction of sound due to neurites from the auditory nerve. Outer hair cells is a cochlear amplifier because they amplify the wave that came through. So that's what the outer hairs do. They help amplify that wave. If you damage the outer hair cell, it causes deafness. So the outer hair cell, if you've ever been to a concert, very, very loud, and then you leave the concert, you hear like a whooshing noise for a while. That's like you overstimulated your outer hair cells from a loud noise, and after a while it takes a little bit for them to reset and go back down to normal. Ototoxicity. Oto refers to the ears. Toxicity means it's toxic. We have some drugs that cause hearing problems. So if you prescribe a drug to a patient and they come back a week later saying, you know, I'm having a lot of trouble hearing now, see what you gave them because aminoglycosides can cause ototoxicity. So that's hearing. Now let's talk about how we have balance and equilibrium. How do we balance things? How do we know we're upright? It requires control of eye movement and body posture. So that's how it tells us what way we're going. Our eyes help keep us on the horizon so we know we're going forward, backwards, up or down. Any vestibular dysfunction gets uncontrolled eye movements, feeling of nausea, and vertigo. So if you ever go on those simulation rides at like Disney or Universal, where you have the visual of what you're looking at, you have the movement of the cart that moves around, you have sound, and you have the vibration in your that's picked up by your senses in your tendons and ligaments for stretch. If the vision is off of what you're moving, you get sick. You get like motion sickness because the sight, your position sense, what you're hearing, all these things have to be coordinated. And if one thing is off, everyone's puking everywhere because your balance gets off. So the sight, the movement of the car, all those things have to match. That's why in the beginning of like all this virtual reality stuff with the headsets, at first they weren't very good because people were getting sick because what they were hearing, what they were seeing, what they were doing was off. It didn't sit, so people were getting sick. They fixed it all. Now they can use VR a lot better now. But in the beginning, that was a problem. It was off. Vestibular cochlear nerve couldn't handle it, and you got sick. So there are two structures that provide our vestibular sensation. They both contain endolymph. So if it's endolymph, what is the ion that does the depolarization? Potassium. We have otolith organs, and there are two of them. There's a utricle and a saccule. And this detects tilting of the head and linear acceleration. So if I tilt my head downwards, how do I know I'm looking down? Because of these organs. How do I know I'm going fast this way in a horizontal acceleration? It's these organs, and I'll explain what they do in a second. The semicircular canals detect angular acceleration. Okay, so we know, are we going on an angle? Are we going up, down, sideways? Which way are we going? And that's where the semicircular canals also come into play. So here are the setup. Utricles are for horizontal motion. Your saccule is for vertical motion. So remember, these are all covered in what? What do these reside in? Like a jelly substance, some fluid that's there. So, utricle, horizontal movement. I'm walking this way. That gel is hitting that direction, and all these stereocilia bend in one direction. That's how I know I'm going horizontal. The saccule looks at vertical changes. So I'm looking down. All the gel pushes down on that stereocilia, bending it toward a certain direction. That's how I know I'm looking in a certain way. Put my head all the way up. How do I know I'm looking up? the saccule bends in the opposite direction because the fluid's moving in the opposite direction. So the utricle, I know I'm going forward because I'm going this way. The gel is moving in that direction. If I go this way, the gel is now moving in that direction, and the utricle bends the opposite direction, and that tells me I'm going the opposite way. Everyone good? If I'm in that ride where you're sitting down and it shoots you straight up in the air and you go up and down a bunch of times, okay, same thing. If I'm going up, all these stereocilia are bending one direction. That tells me I'm going in that direction. As I come back down, the stereocilia bend the opposite way, and it gives me the sensation I'm going the opposite direction. Okay? This gel contains crystals, and sometimes these crystals can precipitate, and they come out of solution, and they cause crystals in the ear, and we call them otoliths. So some people get like vertigo. They have like crystals in their ear, and you have to get them out. So there's different procedures you can do to get these crystals out to help you with your balance. So, like I said before, let's go up this picture right here. Here are the utricle or the saccule. It doesn't matter which one it is. They all work the same way. The big hair cell is called the kinocilium, and all the other little ones are called stereocilia. If the kinocilia bend towards the stereocilia, we get deactivation, a decreased discharge. So I'll tell you you're going in a certain direction. If the stereocilia bend towards the kinocilium, the big one, then we get activation, increased discharge. That tells you again, we're going in a direction. So even though only one of them has an increased discharge, the opposite direction has a decreased discharge, which is also sensed by our brain, so we know what direction we're going in, based off of which way the stuff bends. If the stereocilia bend towards the kinocilium, we activate it. If the kinocilium bend towards the stereocilia, you get deactivation. But what does that tell you? Just direction sense. Am I moving forward or am I moving backwards? That's all it is. You don't have to know which one is which. Just understand. It just tells us direction sense. Okay, that's what I talked about there. So again, like I mentioned before, as you're looking in certain ways, and you look downwards, the hair cells will bend because that gel that's in your ear will cause those hair cells to bend either towards the kinocilium or away from the kinocilium. But the end result is the same. Which way are you moving? The semicircular canals. These help with angular accelerations. Okay, in this case, we have this thing called a cupula, which is right there, which, if you look inside, has all of the stereocilia there. They're all right there. So again, as it's bending in different directions, tells you what angle you're going in. Superior canal, front and back, so nodding your head. This movement is picked up by the semicircular canals. Horizontal canal, left to right movement, so shaking your head. This is your semicircular canal. And posterior canal is rotation and plane from left to right shoulder, so going like this or like this. Okay? So, how many directions can the semicircular canals tell us? 6, or basically 3, right? 3 directions, whether they're discharged or activated, will tell you if it's going one direction or the other, but 3 directions. How many do the otolith canals tell you? Directions? 2. So how many directions do the ears detect? 5. There are 5 directions that the ears will pick up and they will sense. 2 from the utricle and the saccules, the other 3 from the semicircular canals. And that's how we know where we are in position to the world. The equilibrium neural pathways, I talked before about how a lot of things have to be in sync so you don't get sick and throw up everywhere. Okay, and it goes to the medulla oblongata from the vestibulocochlear nerve, and obviously back into the cerebellum. Cerebellum is responsible for what types of movements? Coordinated movements. Okay, if I'm walking and I'm going to hit a wall and I need to turn around, doing this is considered a coordinated movement. Touching your nose, doing all these things are coordinated movements. They all go to the cerebellum. Medulla sends neurons to the oculomotor area of the brainstem that controls eye movements and obviously helps with our body movements. Whenever you hear a sound, your eyes dart to the sound or what side of the body you heard that sound. If you hear a loud sound on your right, your eyes will go to the right automatically. It's all part of this neural pathway that we have set up to keep our balance. So you can see here, cranial nerve gets to the cerebellum, limb motor neurons, cerebellum. Our EOMs are here, your neck motor neurons to look in different directions. So if you hear a noise, you look that way. Again, your neck has to be involved in that. All of these things send inputs to the brain as far as where you're looking, which direction you're looking, and so forth. So if you go on a cruise and you've been on that rocking boat for like 7 days, you're going to get used to the rocking, and you will walk in a certain way so you don't start swaying. And you get used to walking that way. But then you get on land, and nothing is moving anymore. So it's like that drunken sea legs, whatever they call it. Because you're used to a certain way, and you don't have it anymore, so you're like walking into walls and getting off balance. It's because you have to reset your vestibulocochlear pathway. It takes a couple hours, and you're fine. It's like if you ever see people that come back in from like space, they can't even walk for a couple of days. All of it has to do with muscle loss from zero gravity, but also there's knowing where they are in the world. Vertigo is your spinning. Either the body or the environment is spinning around. So there's a mismatch between your vestibular, visual, somatosensory. Gives you that sensation of dizziness. So there's a mismatch there. Something is off, giving you that sensation. And the nausea. What is nystagmus? Anyone know? Eyes start bouncing around. It can be horizontal, it can be vertical, it can be rotary, but there's movement of the eye because something is off with your vestibulocochlear system. Any questions about how you hear and balance? Okay. Gustation and olfaction. What's gustation? Taste. Olfaction. Smell. Very good. So, two most familiar examples of chemoreception. Okay. In the eye, it was photo. Right? Now we're doing chemoreception because when we smell something or we taste something, the chemical that we are smelling or tasting is going to bind to a receptor. And then you get from there. Hundreds of different olfactory receptors. We've lost a lot of our smell sense through evolution. We don't need it. Obviously, like if you have a dog, dogs have like thousands of receptors for smell, because that's how they communicate through smell a lot more than we do. We have words now. There's only 5 basic qualities of taste: sour, sweet, salt, bitter, and umami. Those are the only 5 things we taste. Just like we did with the eye. If certain red, green, blue things are saturated, you're going to get a certain type of color. With your taste, we taste all of these things, and it's very, very salty. We'll taste more of the salt in there. If you add a lot of sugar to the food, there's still salt there. There's still other things, but the sugar is going to overpower the taste to give you more of a sweet taste. So it's all perception, and smell and taste kind of go together. That's why when you're sick and you have like sinusitis or a nasal infection, you can't taste food very well because the smell receptors are all occupied. They don't do anything. So our gustatory receptors, they're little taste buds on our tongue. Each papillae have numerous taste buds which open to the surface of the tongue, creating a pore to allow whatever we're eating so we can taste what we're doing. So you can see we have our taste pore. There's the taste receptor cell, and that's going to convert it into a electrical signal. that our brain can then detect. So, how do we taste different sensations? Salt makes sense. Sodium. You add sodium to your food, the salt receptor, which are just sensing for sodium in the food, will be activated and you will taste salty. Sourness is a response to acidity, therefore it is hydrogen. Hydrogen. Anyone here have any Sour Patch Kids? Or anything sour? No? Okay, sometimes people do. The main ingredient that makes them sour is acetic acid. That's it. Acetic acid gives you the sour taste. And you're testing for hydrogens. That's what gives you the sour sensation. Sweetness uses intracellular second messengers. Whenever you see this word secondary messenger, we haven't gotten into them very much. We'll get to it a lot in endo. That's why I see chapter 8. Secondary messengers, all they do, amplify the signal. So if you ever see secondary messengers, G proteins, they amplify the signal that was given to that cell. Bitterness, a bunch of different mechanisms, but the majority is potassium channels and changes in secondary messengers. Bitterness is something we do not like. It is an evolutionary safety mechanism, so we don't eat bad food or bad mushrooms or whatever. If we taste something bitter, we spit it out. Okay, so our bodies are made, if we have something bitter, we don't eat it. That's why a lot of those little button batteries, they coat them in a very bitter solution. So if a kid goes to taste it, they taste something really, really bitter, they spit it out, they don't swallow it. So that's why all those batteries are coated now. So that's what bitter taste is. It's just potassium. It's a mechanism we don't eat these things. The whole cilantro thing. Anyone here have that cilantro taste like soap? It's a genetic thing. Your genes are coded that way, and that's why you can't taste cilantro tastes like soap. It tastes disgusting. It's a bitter taste because of the way your tongue is. The last one is umami, which is just Japanese for delicious. It responds to amino acids, particularly glutamate, which is your MSG. What is glutamate? It's a neurotransmitter, mainly in the brain. What is it, inhibitory or stimulatory? Stimulatory. Glutamate is the major stimulatory neurotransmitter in the brain. Okay, so umami, delicious flavor, is glutamate. You respond to amino acids. So now you know how we taste food. What are the receptors for each type of flavor? And again, the more receptors are occupied with certain flavors will give you a flavor profile. That's how we have different tastes. So, taste pathway. What actually tells our brain what we are tasting? That facial nerve, cranial nerve 8. Okay? First 2/3 of the tongue. So the first 2/3 of the tongue, the facial nerve will take that signal to the medulla oblongata, then up wherever it's got to go. The last third of the tongue is the glossopharyngeal nerve, is the posterior third of the tongue. And that's how we sense taste in the back of our tongue. It's the glossopharyngeal. So if someone had a facial nerve paralysis, would they be able to potentially taste things? Not very much, because the front 2/3 of their tongue is supplied or sends the signals via the facial nerve, and the facial nerve doesn't work, you're not going to taste on certain parts of your tongue. Again, this is sensory, so all sensory has to go through the thalamus. Okay, and then the thalamus, it goes and branches off from there, and it goes to wherever it's got to go for sensory of taste. You don't need to go crazy with this one, just where it happens. Big thing is know the different receptors for taste and know the innervation of the tongue that allows us to get that taste to our brain. Salivation. Okay, we have salivary glands. that allow to moisturize our food as we're eating. Okay, if you know you're going to be eating pretty soon, it can cause a salivatory reflex, and your mouth will start watering because you're preparing to eat food. If you go to Chili's and you smell the fajitas going across, you start thinking, oh, I'm getting fed pretty soon, your mouth starts watering. We have the sublingual gland, that's innervated by facial nerve. Submandibular gland is also facial nerve, but the parotid gland is the one that's different. That's the glossopharyngeal. So just remember, parotid is glossopharyngeal, all the rest are facial. You don't worry about anything else to study. You know the one odd one, everything else is the facial, you're good to go. So again, we taste something, we get salivatory response to that to get ready to start eating. Swallowing, you do not need to know this, but the point of this is there are 6 nerves that are just needed to swallow. So you to coordinate swallowing requires the movement of, or the integrity of 6 cranial nerves. The reason why I bring it up is because anyone that's coming in with a stroke, you don't know if any of these nerves have been damaged in the stroke. So that's why all stroke patients, you keep them NPO, no eating. Because there's so many nerves that are involved in just the act of swallowing that if someone can't swallow properly and you give them food, they're going to choke on it, they're going to cough, and what's the worst thing that could potentially happen? Aspiration pneumonia. Now you've breathed in that foodstuffs that should not be in your lungs because of all of these issues with these nerves. Again, you don't need to know what nerves are involved in swallowing, but just understand that there are a lot of nerves involved, very complex, and be very careful with swallowing. Olfactory receptors. These are also bipolar neurons. So olfactory and the retinal are the 2 ones that are bipolar. And they project into the nasal cavity. This is the only part of the brain that's really exposed to the environment, which is why you gotta be careful picking your nose. Because if you have an infection on your fingers and you introduce it into your nose, it can get up there and start activating these little receptors and cause an infection. Okay, so I'm not saying it happens all the time, but be careful. Okay, so that's exposed to the air. Again, cilia bind to the odors. Okay, we smell it. We have about 380 genes. Obviously, animals have a lot more than that because they use that for communication. We don't anymore. One odorant molecule will stimulate one protein. So it's basically a 1-to-1. Okay, and then you can see down here, here are the odorant receptors. Here is the air that comes in with perfume or whatever's going on. And when you smell that, that perfume molecule will go into your nose and it will bind to a receptor, and then you get the sensation of smell. Through cranial nerve, which one is your smell? 1. Okay. Olfactory are these G protein coupled. Just remember, anytime you see this G protein, secondary messengers, it just amplifies the signal. That's all you gotta know for right now. And again, membrane depolarization occurs. Here is the odorant, and here are the calcium channels, and we work our way up, and then we go up to the brain, olfactory nerve, and we can sense what we're smelling. So the big thing with this one is just know one molecule binds to one receptor. It's a chemoreceptor, and it tells our brain what we're smelling. So taste and smell together. We have interoreceptors, so they can make changes in what's going on. That's within the body. We have exteroreceptors. These are from outside the body, include taste and smell. Taste responds to chemicals dissolved in food and drink. Smell is the chemicals from the air. So when you get a bowl of soup or whatever, the molecules that are coming off of it are detected by the nose, and you smell it, and then you taste it after that. So that's why olfaction greatly dictates or influences your taste. So again, taste and smell, they both go together. We have our sweet and taste centers. They both kind of converge in that one smell center from the nose and the tongue. so our brain knows what we are smelling and eating. Moving on to our autonomic nervous system. Let's take a break, come back at 2:30. Okay, let's get going. We don't have that much left today, so we will get out early again. So, the last section is just on sensory. How do we perceive the outside world? Taste, vision, hearing, touch. So when you're studying, think of that in mind. How am I sensing something? How does a pathway go from, if I'm hearing something, how does that sound go? Through the ear, the outer ear, all the different parts. And as you're doing that, you can study your anatomy, because you got to do all that anyway. So study the anatomy. What parts are going through the ear? Okay, now I go through the inner ear, and then the outer ear, or outer ear, then inner ear. Like, what are the structures I'm going through? That helps you study for anatomy. So bring other courses in when you're studying this stuff. So that was all just on sensory. How do we perceive the outside world? Now we're going to go through more detail about the autonomic nervous system by itself. We'll talk about sympathetic and parasympathetic. Our autonomic nervous system is throughout all of our visceral organs, cardiovascular system, and exocrine organs. Without conscious awareness, it just happens automatically. We don't have to worry about anything. What's the purpose of it? Maintains homeostasis. That's the whole point of this whole entire course, is learning out how our body maintains homeostasis. Integration of stress response. That's our fight or flight response. So when you're studying this thing, always remember sympathetic, fight or flight. Parasympathetic is your rest and digest. So right now, your parasympathetic system is in charge. You're resting, you're digesting the food you have for lunch. Me, I'm up walking around, a little more active. My sympathetic is probably kicking a little bit more than yours are. If you are stressed out right now for some reason, your sympathetic system kicked in, because you're causing a fight or flight response. So the divisions of our autonomic nervous system: sympathetic, parasympathetic, and our enteric, which we'll cover in GI later on. It's closely integrated with our endocrine system because of the hypothalamus. Okay, the hypothalamus tells the pituitary gland what to do and controls our whole body with our hormones. Hypothalamus is involved with that. In general, sympathetic and parasympathetic are like opposing sides, like on a seesaw. One may be in charge than the other one. Normally, parasympathetic system is in charge all the time. When you need sympathetic, then it kicks in. So sympathetic activation due to response to stress. When you're thinking about this, just think, okay, if I'm going to be fighting a bear, okay, what am I going to have to do to survive, which is nothing, but let's pretend that you could. Cardiovascular system. Heart rate and contractility increases. Heart rate goes up. Why? I got to pump more oxygenated blood through my body. I got to get more glucose through my body. The way you do that is pick up the cardiovascular system. Blood redistributed to the muscles, heart, and brain. Muscles, yes, I want my blood to go to my muscles so I can run away, so I can fight, so I can do something. I need the blood in my muscles so I can do everything. Heart, brain, so obviously keep functions working, keep you alert. Tissue metabolism during a sympathetic activation. Energy utilization by glycogenolysis by the liver. What is glycogenolysis? Into glucose. Makes sense. I need a lot of energy to do whatever I'm going to be doing against this bear or whatever. So I need sugar. I need glucose. I need an energy substrate. So the liver starts dumping out glucose into the bloodstream. Smooth muscle. Contraction of sphincters of the GI and urinary tracts. You do not want to have to stop and pee if you're running away from a bear. So by having all these sphincters tightened up and closed, it prevents you from having to stop and go to the bathroom. So it allows you to close all those areas so you can keep doing what you got to do. And dilation of bronchioles. Why do you think we want our bronchioles to be dilated? What are bronchioles? Airways in our lungs. Why do you want those dilated and as big as possible? More oxygen into the body. You breathe more. We're going to need a lot of oxygen. Let's do a lot of breathing. We'll get a lot of oxygen going. Dilation of the pupil. Remember, we just talked about this before. Pupillary dilation is under what control? Sympathetic control. Our sympathetic system is now activated. We have pupils that are going to be wide. Why? More see, you can see better. More light coming in, you can see your surrounding easier, and you can fight off an attacker. So this is what happens during a sympathetic activation. And always think, I'm going to be fighting or running away from something. I need lots of blood to my muscles. I need lots of oxygen. Heart rate goes up. Breathing goes up. All these responses allow me to do whatever it is I need to do to run away. Parasympathetic activation. Conserve and restore energy reserves. Heart rate goes down. Don't need a fast heart rate. We're not doing anything. So the heart rate comes down and relaxes. GI function is increased. Now we can start digesting and moving things through our GI system so that we can absorb the nutrients. And that all happens at the parasympathetic level. Sympathetic nervous system, the way it's set up, we have what are called two paravertebral sympathetic chains of ganglia. Also, you may also see it as a sympathetic side chain ganglia. What am I talking about? It's all of this. These are all ganglia, collection of cells, right off of the spinal cord, the sympathetic side chain ganglia. And then you see they move out. Sympathetic innervates the eye, the heart, bronchi, liver, stomach, through the celiac ganglion. We have our intestines, kidney, colon, all for those sphincters, okay, to control everything so we can move stuff around. Okay, that is the sympathetic system. Okay, nerves and extending from the ganglia to the internal organs to have their effect on those organs. This is it for the sympathetic system. This is all the things that it does. Nothing else. Doesn't do anything else, just these things. And as we go through different systems, you'll see how the sympathetic system plays a role in doing certain things. And we're going to get to a section that has of all the receptors, and I'll tell you which ones I want you to know for your exam. So the sympathetic nervous system, we have this ganglion, the side chain ganglion next to the vertebra. Okay, now, when we have the ganglion, this is where it is right here, the sympathetic side chain ganglia. This is the dorsal. Here's their ventral root. What does the dorsal root convey? What type of information? Sensory. Sensory has to go towards the what? Brain. So if you look at the arrow, the green arrow, you see how it's going into the spinal cord? That's how you know it's sensory, because the pathway are going into the spinal cord, up to the brain, and then we can do whatever we need to sense from our big toe. You know, if you step on a tack, it's going to travel these ways. The sensory has to go into the spinal cord and then up. And if you look at the ventral root, what is the ventral root? Motor. That's got to leave the spinal cord to go to your extremities to move something. So if you look at the red is going out. It's leaving the spinal cord. So that's why, always remember, The dorsal is always sensory. Ventral is always motor. So all we're saying here is that when it comes into the ganglia, there's three things it can do. It can go up, it can go out, or it can go down. That's all we're saying here. When it gets to the side chain ganglion, the impulse has three different ways it can go. It can stay within the sympathetic side chain, go up or down, or it can leave at that level. That's all you really got to know for this stuff with regards to sympathetic. As far as which direction it goes, does it stay in the ganglia? Does it leave? Those are the only two options it has. Acetylcholine is the primary neurotransmitter released by the preganglionic neurons. Okay? So, what does that mean? This is leaving the spinal cord, and here is our ganglia right there. When a neuron, sympathetic neuron, leaves and it gets to this area, it's going to have a synapse. Put an arrow like that. And it's going to release acetylcholine. That acetylcholine is going to bind within the ganglion, activate the secondary nerve or the postganglionic nerve, which then goes to an organ. It's going to have a different receptor there. So, acetylcholine is always preganglionic, nicotinic on the postganglionic neurons. Norepinephrine is the primary neurotransmitter released by the postganglionic neurons onto target tissues. The only exceptions is the sweat gland and the adrenals. They have different ones. We'll get to that in a second. The target cell, where it's going to go next, is an adrenergic receptor to norepinephrine, and these are your alphas and your betas. So remember, if you ever see an alpha receptor or a beta receptor, it's always going to be sympathetic system. Alphas and betas are always sympathetic. Do I have a picture? No, I don't. Let me draw a new one here. Okay, so this pregang, postganglionic. Okay, acetylcholine, nicotinic. But at the post side, it's going to be norepinephrine. So when it gets to the postganglionic, at the very end, the terminal boutins there are going to release norepinephrine. It's a sympathetic nerve. But the preganglionic is always going to be acetylcholine binding to that nicotinic receptor. That sends the active potential down that way. Now we're in the postganglionic nerve. When we get to the end of that one, because this is sympathetic, it's going to release norepinephrine. So I kind of just drew it out. I'll have some more graphs in a little bit later on that kind of explain it as well. But just so you know, I'll put SNS down here so we know that it's sympathetic nervous system. In the sympathetic nervous system, the preganglion nerve is very short. The postganglionic in a sympathetic is very long. Why is the preganglionic in a sympathetic nervous system, why is that nerve short? Where does it have to go to? Where is the ganglion that it goes to? Right next to the spinal cord. So it's a short nerve, it doesn't have to go very long. But then after the ganglion, there's a long nerve, and I'll show you this picture here. Well, you can kind of see here. Let me erase some of this stuff. Okay. Very short here, right? Very short. And now the postganglionics are very, very long. Okay? That's what I'm referring to. In sympathetic nervous system, the preganglionic nerve is short because it only has to go to the sympathetic side chain ganglion, which is right next to the spinal cord. Once it synapses there, the postganglionic nerve is very long in sympathetic systems. The adrenal medulla is an endocrine gland at the core of the adrenal gland. We have sympathetic preganglionic neurons that go into this area that stimulate the medullary endocrine cells to release epi and norepi. Okay, this is a neuroendocrine response. So it's mediated by the neural system. It's going to tell it to release norepinephrine and epi very, very fast. Why is that important? That epi and norepi come out very, very fast via the nerves and not like in an endocrine system where it's blood and you have to wait for the hormone to travel throughout the blood and finally get to the target system and have a response. With the adrenals, it's fast. Why? It's fight or flight response, our sympathetic system. We don't want to wait, like, oh, I got to wait a half hour before I can fight you. No, we got to do it right now. We got to go. Okay? It's a neural mediated response. It's going to be fast. If I know I have an anxiety attack, it kicks in 2 seconds and you start breathing faster, all these other things. Heart rate goes fast. Look at the sympathetic responses. Heart rate goes up, breathing goes up. An anxiety attack is just a sympathetic response. That's all that's going on. So epi and norepi come out. The actions of epi are complementary to norepi. We'll get into that a lot more detail when we get to the endocrine system. But again, these are sympathetic hormones. These are adrenergic. So we're going to do the alphas and the betas. So that is the sympathetic system and how that is set up. Let's talk now about the parasympathetic system and how that is set up. There are only a few cranial nerves that have parasympathetic input. 3, 7, 9, and 10. Those are the cranial nerves, and that's it for parasympathetic. And then we have some at the bottom too, through our sacral nerves here at the bottom as well. But those are the two areas that have all the parasympathetic innervation leaving the spine. Okay? So, you can see here in this picture, let's look at the ganglion and where they are. Okay? Preganglionic fiber for parasympathetic is very long. Postganglionic is short. It's the opposite. In the parasympathetic system, the preganglionic nerve is long, the postganglion neuron is short. Because the ganglion for the parasympathetic system are out in the periphery. We have a ciliary ganglion, pterygopalatine ganglion, which helps with salivation, and more salivation glands. With those ones, so these are more lacrimal and nasal ducts, I'm sorry. Heart is involved, your stomach is involved, and your sphincters are here as well. Parasympathetic is what? Rest and digest or fight or flight? Rest and digest. So you can have the time to go to the bathroom. So you do at some point want those sphincters to relax so that urine can pass and you can go to the bathroom. So this is the parasympathetic system. Very, very few nerves that come out. This is what I was talking about with this whole long and short. This kind of goes to a little summary slide, a great review slide. The top one is the only one that's parasympathetic, sympathetic, 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 and then your adrenals. You don't need to know right now that like these two are different, but if you look, This is your parasympathetic, and at the end, what is it? Acetylcholine onto muscarinic. Acetylcholine as the final neurotransmitter means you're a cholinergic neuron, and that's found in parasympathetic systems. Sympathetic, norepinephrine, and it's going to bind to your alphas and your betas. So here are your alpha and your beta receptors. Heart and your vessels. In the sweat glands and vessels as well. Even though it's sympathetic, it's acetylcholine onto your muscarinic receptors. I mentioned acetylcholine binds to nicotinic and muscarinic receptors. Where are nicotinic found? Skeletal muscle. Your muscarinic are in your sweat glands and your vessels. So when you start running outside, it's Florida, you're outside for 2 seconds, you start sweating already, it's your sympathetic system kicking in to allow your sweat glands to contract and push sweat out to your body. In your renal system, it's dopamine is the neurotransmitter. And dopamine will bind to its dopamine receptors, D1 or D2. And then here are your adrenal glands. We said, remember, the adrenal glands are neurally mediated. So as soon as this nerve hits it, it dumps out norepinephrine into the bloodstream, which will then go to the body to activate your betas and your alphas, kicking in that sympathetic response. So this is just a summary slide talking about the different setup for sympathetic versus parasympathetic. Receptor effects. What happens when they're bound to the receptors with these autonomic or sympathetic or parasympathetic? Excitation or inhibition, depending on what type of tissue we're talking about. Receptors include ion channels, which is your calcium, potassium, sodium. Those are your ion channels. Or metabotropic effects. This is your secondary messenger, your G proteins. Remember, G proteins are going to amplify the signal. Cholinergic receptors, we have nicotinic, ligand-gated ion channels. So obviously the acetylcholine is going to bind to the nicotinic receptor. Then we have opening of the channels. Muscarinic is here as well. And then you have your adrenergic receptors, alphas, and your betas. Now, this picture here, this graph, there's a couple things I want you to know, and I'll tell you which ones. The heart. And let's look at just sinoatrial node. The sympathetic response is to increase the heart rate. How does it do it? Beta 1 receptors. So to increase your heart rate, you have to activate your beta 1 receptors. The parasympathetic response is the opposite, decreases the heart rate. All mediated via muscarinic receptors. Why? It's autonomic. If it was somatic, then it would be nicotinic. But we're not somatic. So, sinoatrial node with sympathetic response through beta 1 activation, you're activating those beta 1 receptors, heart rate goes up. The next one I want you to know, bronchiolar smooth muscle. Relaxation is caused by beta 2. Okay? Remember, 1 heart, 2 lungs. Beta 1 is in the heart, beta 2 is in the lungs. Easy way to remember that one too. And the parasympathetic response in the bronchial smooth muscle is to cause constriction. Okay, I also want you to know, okay, urinary bladder, detrusor. The detrusor muscle is the muscle that actually, of the bladder, that squeezes to allow you to avoid urine. So, the sympathetic system causes relaxation of the wall. We want to relax the wall, and what will that allow you to do? Hold more urine. Because we're relaxing the bladder, it can hold more volume. If the bladder was constricting, you would go to the bathroom. But going to the bathroom is a parasympathetic response, not a sympathetic one. So if you look at here, contraction of the bladder wall is a parasympathetic response. It allows you to void so you can go to the bathroom. Okay, so you remember I told you it doesn't matter how stupid these mnemonics are? I'll give you an example right now, easy to remember. Genitalia. Ejaculation is sympathetic. Erection is parasympathetic. So the way you remember that is point and shoot. The pointing is parasympathetic. The shooting is sympathetic. And now you know how that works. Easy way to remember it, doesn't matter how stupid it is. Point and shoot, erection, ejaculation. Now you know what systems are involved. That's pretty much it. I want you to know, PAs, you're going to get all this with Dr. Wood and Farm. You're going to learn it anyway. You're going to get it anyway. Start associating the receptors to the system that they belong to. Always remember, nicotinic muscarinic is acetylcholine. Beta 1, beta 2, beta 3, alpha 1, alpha 2 are going to be your sympathetic ones. Start getting those in your head so you know when we talk about sympathetic activation, we know it's alphas and betas. Parasympathetic activation, muscarinic, nicotinic. Okay, the last little bit we'll talk about today is just integrative and behavioral functions. How do we remember? Memories, anxiety, fear, depression, all that fun stuff. 3 areas of the brain for implementation of integrative function. This sets us apart from most of the other animals in the kingdom. Hypothalamus, major controller of endocrine and autonomic nervous system. A lot of our homeostatic variables are sensed by the hypothalamus. It also deals with eating, circadian rhythm, and sex drive is all based off of the hypothalamus. Whenever you see the word reticular formation, think of level of consciousness and arousal. That's what the reticular system is for. And we also have the limbic system. And the limbic system is where our emotions they're stored. Our emotions are here. It has reciprocal connections with the reticular formation and the hypothalamus, which is why when certain memories that you have in the hypothalamus will evoke a certain response, you may have an emotional response to that as well. Even though it's all in your head, you're thinking about it. Because the limbic system talks to the reticular formation, hypothalamus, you'll have a response to that. So hypothalamus, area below the thalamus, has a bunch of nuclei. You don't need to know all this stuff. Don't worry about the nuclei. Hypothalamus has afferent inputs. Afferents, do they go away from the hypothalamus or towards the hypothalamus? You sure? Away. Wait. No, afferents are towards. Efferents are away. Yeah, sorry. So it has afferent inputs. It's knowing what's going on in the outside world, in the environment where we are. Also has our open area here for the blood-brain barrier in the hypothalamus. We can sense our osmolarity of the blood, what the concentration of our blood is. With solutes, the hypothalamus can detect all that. The frontal lobe and part of the limbic system link the higher centers of mood and emotion. Your frontal lobe is your mood, your personality. That's who you are. So if you ever hear of a frontal lobotomy, what they do is they go through your nose and they scramble the frontal lobe so you don't have those crazy personalities or outbursts or whatever it is. That's what a lobotomy does. Also has efferent connections, so descending pathways via reticular formation. Again, arousal, sleep, level of consciousness. Output to endocrine system, and also your limbic system. So it has these efferent pathways that leave the hypothalamus. Food intake. We have 2 parts of the hypothalamus that regulate our food and eating. We have the ventromedial nucleus is our satiety center. What is satiety? Feeling full. Okay? Our feeling of fullness is in the ventromedial nucleus. Our feeling of hunger, feeding center, is the lateral hypothalamic area. So these two areas sense when we're hungry and when we're full. Neurochemistry is complex. We have orexigenic type of things which stimulate feeding. And we have anorexigenic, which inhibit feeding. So anorexia, we're going to inhibit feeding. These are with your corticotropin releasing hormone and the CART system, cocaine and amphetamine pathway, which is why people go on cocaine for a while. They get very, very thin because they're not hungry. Because the cocaine system inhibits feeding. They're not going to be hungry. That's also, I think, where Ozempic comes into play. Ozempic tells your brain that you are not hungry. So therefore, you don't eat anymore. Because that sensation of hunger is gone. Because you're tricking the brain into thinking that you're full. So you don't eat much. So that's the different types of hunger. Regulation of food intake. Short-term factors. Hypoglycemia. Your sugar level drops, you get hungry. Sugar goes down. There's also a hormone called ghrelin. Okay? That also tells the brain, hey, we're eating. We're hungry now. Let's eat something. After you eat, the sensation of satiety is sent via the vagus nerve to release what's called cholecystokinin. Don't worry about it now. We'll get to it when we get to the GI system about that. The long-term factors that tell us our fat stores, the hormone is called leptin. Leptin is secreted by our adipose tissue. So the more adipose tissue you have, the more leptin you have. This also has a role with long-term feeding. July 4th is coming up. What happens in July 4th every year in New York? Up in Coney Island. The what? Is it the hot dog eating? The hot dog competition, right? They got 10 minutes to eat all this food. The problem is, for those first 10 minutes, you can eat a lot. Because the brain hasn't picked up yet, hey, you're eating, you can stop now. So what happens is for those 10 minutes, they can shove as much as they can. But a few years ago, they had to do they were tied at the end. So they had to wait 10 minutes and then eat another round of hot dogs, whatever. But by that time, you've eaten all this food. Now your brain's like, okay, you're full. You don't want to eat anymore. You're not going to want to eat. So that's when you start getting sick and throwing up, all that stuff. So your brain, it takes a while. That's why I say eat slow. Really chew your bites. Because the longer you take to chew and eat, eventually your brain's going to kick in okay, you've eaten enough now. You're full. You don't eat anymore. And you're okay. But if you eat very, very fast, you can trick that and eat a ton of food in a short amount of time. And then your brain's like, hey, we're full now, but you've already eaten whatever. So that's feeding. Those are what's involved with how we get hunger. Body temp. Negative feedback by the hypothalamus. Body temp is stable when the generation of cell metabolism equals the rate of heat loss. Our bodies are not very efficient at making energy. The majority of when we burn ATP, it's just given off as heat. Not energy, we're not very efficient at all. Other animals are a lot more efficient with energy than we are, but we use that to get heat. So when we're cold, what do? Shiver. Okay? You activate your muscles to produce heat. Because we know that when you activate our muscles, most of it gets heat. There's different ways to lose heat as well. Radiation, conduction, convection, whatever. Regulation of our body temperature. We have peripheral thermoreceptors, and they anticipate any threats to our body temperature. We learned about our cold and our hot sensations that we can perceive from the environment. So our body will know, hey, this is really hot, or we're walking out into the heat. We start walking out, we get the sensation of the heat on our skin, the temperature change, and if we're still in it, then what does our body do with all that heat? You start to sweat to cool yourself off. You don't think about it, you just do it because the thermoreceptors in your skin pick up the heat. We got to cool ourselves off, so we sweat. So effectors for body temp regulation, we have our skin circulation, regulated by adrenergic sympathetic tone. So our sympathetic system is the one that controls sweating. It makes sense. We have a sweat gland. That is a sympathetic system. Vasoconstriction minimizes the heat loss to cold. So when you're very, 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 very cold, we have vasoconstriction in our vessels in our extremities so that all the blood, the warm blood, goes to our core and keep us warm in our core. Our body does not care about your fingertips. If you're freezing, they care about your inside. Don't care about your fingertips. You'll lose them. But again, that's why they go away, because you're getting rid of all the blood in your fingertips to keep your core warm. We have vasodilation when the body is too hot. Your skin gets red. Why does your skin get red? You have vasodilation. Capillaries start to leak, and you get red in your skin. You're trying to cool yourself off, bringing more of the blood to the skin so it cools off. You can sweat, which also cools off the blood. Metabolic rate. How many calories does your body burn while you're sitting at rest doing nothing? That's your metabolic rate. How much calories does your body need just to exist is your metabolic rate. If you have a low body temp, increases cell metabolism via your thyroid hormone. Shivering occurs at low temps, less than 95 degrees, but is a short-term response. So shivering is a short-term. You can't shiver forever. Short-term, then it goes away. Sweating is obviously a most important response to body temp, and it's cholinergic, post-ganglionic, sympathetic, which is a review of that other slide from well before. This is talking about how we generate a fever. So I want to walk through this because it's a good review slide. It kind of talk about different parts of the brain, what's open to the blood-brain barrier. This won't be on your exam, but kind of just listen. This will tell you guys why you get a fever, why you break the fever, why do you have sweats when you break it. So fever is a regulated increase in temp in response to an infection or a disease. One of the worst things you can do is giving someone who has a fever Tylenol, unless they're like really symptomatic from the fever. Why is that bad? Our body's response is to raise our temperature to kill off the bacteria and the viruses. They can't survive at high temperatures. So by raising our temperature, we're burning them out. They can't survive. So we need to get a fever to raise our body temp so that we can kill off anything that's inside of us. So the first thing that happens, we get cytokines that are released by the immune cells in response to an infection. So these cytokines start coming out. One of them is called a pyrogen. What does pyro refer to? Fire or heat. Gen is to generate, so we're generating heat, pyrogens. Okay, these second phase, the cytokines target that OVLT. And remember that OVLT is one of those circumventricular organs that's outside the blood-brain barrier. So it's going to sense all these cytokines floating in our bloodstream because it can sense outside. So we start generating prostaglandin E2, PGE2. And prostaglandin E2 raises your body temperature. That's what goes to the hypothalamus and tells it, hey, let's kick up the temperature. And actually, that's what a lot of those NSAIDs do. They block the production of prostaglandin. Therefore, you don't get a fever. That's all it is. So there you go. NSAIDs work at reducing fever because they block the formation of PGE2. You're not helping yourself with like Motrin or ibuprofen. You're just blocking PGE2 from being made, and you don't have a fever. As fever is generated, the body temp is now below the new set point. What's normal body temperature? 98.6. Well, you're sick now, you have an infection, so your hypothalamus is like, hey, let's crank it to 101. That's our new set point. But what is our comparator now? 98. So when a fever first starts kicking in, you ever get chills? You start shaking at first? What you're doing is your body's like, hey, our body is too cold compared to our new set point because we're sick. So you start getting chills. That first little bit, then the chills go away, and now you have a fever. So that's the first part of the fever. When a fever breaks, okay, when it breaks, now your body temperature is 101 because that's where your hypothalamus wanted it. But now your infection's gone. You did your job. There's no more cytokines being released because all the infection's gone. So nothing's stimulating the hypothalamus to do anything. So the hypothalamus is like, okay, let's go back to our normal set point of 98.6. But what's your body temperature at right now? 101, because you had a fever for so long. So what does your body do to cool off from 101 back down to 98? Sweat. So that's why when your fever breaks, you wake up in a pool of sweat because the infection went away. We're resetting our body temp. We have to cool ourselves off compared to what our temperature is now. So we start sweating, and that's how you cool yourself off after the fever breaks. Okay, so now you know how you get a fever and how it goes away. Circadian rhythms. Okay, we have a regular variation in our temperature and hormone levels through a 24-hour period. Cortisol is the big one. Cortisol peaks in the morning. Cortisol is our stress hormone. Why do you think it peaks in the morning? Wake you up. Okay, when cortisol comes out, glucose starts going up in the blood, and you start getting aroused, and you wake up. Growth hormone is the opposite. It actually peaks during sleep. Okay, that's why a lot of those the weight trainers, whatever, they sleep a lot because that's when they get their growth hormone. They don't sleep, they're not going to get growth hormone, they're not going to put any muscle on. So that's why sleep is so important. And all these things contribute to jet lag because if you're used to 8 o'clock in the morning, 7 in the morning, that cortisol spikes, and you go to California, even though it's 4 a.m., it's still 7 a.m. according to your brain clock, and you'll wake up at 4 in the morning with jet lag. Our reticular formation, remember this is our arousal, so I have to go through our reticular formation. There are 4 major systems when it comes to our reticular formation, and it all depends on what the neurotransmitter is that's being used at that time. Serotonergic system, sleep-wake cycle, mood and emotions. What neurotransmitter do you think is being used here? Serotonin, serotonergic. Noradrenergic system. What is this? Is this fight or flight, rest and digest, activation, stimulation, inhibition? What is this one? Fight or flight. This is your stimulatory one. Okay. Alarm center. Most active when a new environmental stimuli is present. So something new, you haven't experienced it before, you're a little alarmed, you're like, oh, like you had an exam this morning, it's your first exam in PA school. AA, you'll have your first exam. You will have a noradrenergic response because it's a new environment, it's a new stimuli, you haven't had it before, you don't know what to expect, so you start worrying. Dopaminergic, okay, this is throughout our CNS. We have a nigrostriatal system. Nigrostriatal, substantia nigra, that's all going to be about movement, voluntary movement. This is one part of the dopamine. The other part of the dopamine is your mesocortical limbic. It's got the word limbic in it. Limbic is our emotion center, and it's our reward system. Okay, you got a great exam score on your exam. You feel great. That is your dopaminergic system, mesocortical system kicking in, giving you a boost of dopamine because you did well on your exam and you feel good. And the last one is our cholinergic neurons, widespread throughout the CNS, and involved in the ability to direct selective attention to a particular task. Okay? So your cholinergic system allows you to focus on your studying while you're learning without any distractions. It's your cholinergic system that allows that. EEG, the electroencephalogram, just measures brainwave activity the same way an EKG measures electrical activity of the heart. Same thing, electrical activity, we can measure it. Measures cortical activity, which is obviously the cortex, where a lot of our motor and sensory are. They can be categorized. What I want you to know big time is that the delta wave is your sleep. Okay. So delta, when your brain gets into a delta wave, that means you're in deep sleep. So here's like a sleep again. Obviously, serotonergic, noradrenergic systems are very active in our sleep-wake cycles, help with waking up. Our non-rapid eye movement or non-REM sleep, parasympathetic system is active. And then we have a couple of different stages. REM sleep is dreaming sleep. The brain is awake, but the body is paralyzed. Does you ever hear those sleep paralysis? That's what's going on. Your brain's awake, you're dreaming, you can see things, but your body can't move. You're still like in that deep sleep, but you kind of woke up. So that's sleep paralysis. Here, you can see, again, slow, stage 4. Slow wave sleep is your delta waves. Those are your deep sleeps. That's how we know we're there. Okay, the limbic system. Cingulate cortex. Again, definitions. These are sensations of emotions are perceived here. So you have a new emotion, your cingulate cortex is going to perceive that emotion. The hippocampus is responsible for converting short-term to long-term memory. Hopefully all your hippocampuses are working very well, because you got to convert a lot of this stuff into long-term memory and get it in there. The amygdala is our strong emotion center, fear and aggression. And this links emotions with memories. Give an example. If there's certain smells that may bring like your childhood back, that is acting on all these systems here. It's a memory that you've had in the past that you are now remembering, and you can link what you were feeling. Like when you listen to a song, you may remember of something that happened years ago, or bring back certain memories. One tip for studying, and this is a way to kind of hack your brain. If you listen to music in the background, which is fine when you're studying, the problem is when you come to an exam, it's dead quiet. And for some people, that may make you anxious just having that quietness. A way to trick your brain to really study well is for each class you study, have a different like scent that you have in the room. And the morning of that exam, use that scent again. Helps bring back the stuff that you learned because you were learning it while you were smelling that. The same way here, you can bring back that smell, and it'll bring back memories of maybe things that you learned when you were studying. So scent matters, yes?
Is that the same thing like when you chew gum, when you're studying that you chew gum?
Maybe. I don't know about memory. That's more like just doing something else while you're studying. Okay, a couple of big things here. Prefrontal cortex, association cortex, okay? Memory processing. You see how it goes back and forth between the hippocampus and the prefrontal? So what does that mean? Is that every time you get exposed to something, your brain remembers what happened during it, which is why if your brain is always like in an anxiety mode, your brain will automatically default to that because you had a bad experience once, and now every time that experience happens, your brain is like, oh, this bad thing is going to happen every time. And that's what starts off like the anxiety, because your brain's like, hey, this happened before. I'm going to do it again now. Am I going to have the same response or am I going to have a different response? And that's where your frontal lobe and your hippocampus are talking to each other. You're comparing a experience you had once, because now you're having that experience again. And if you had a bad experience the first time, your brain will default, hey, this is going to be a bad experience. Your anxiety starts ramping up. And what happens is you have to relive that experience twice. Once in your mind, like if you're anxious about your exams, PAs, you're going to have about 120 exams until you're done. Okay, so if you start getting anxious now, every exam, it's like, it can be a different response, but if you start getting in your mind like, oh my God, I had one bad exam, the next exam I'm going to be bad too, you see how your anxiety will make you live the moment once, and then live it again when you're taking the exam. So work on your mindset and get rid of that stuff. Expressions of emotions is a hypothalamus. Also goes to our brainstem and spinal cord. So if you ever had an anxiety attack, hypothalamus tells the brainstem, your respiratory rate goes up, your heart rate goes up, all part of the anxiety response. There are only 2 fears that we're born with. Everything else is learned. Anyone know what those 2 fears are that we're born with? Not the dark. That's snakes. It's height, and loud noises. So like the thunder that goes crazy, that's like one of our innate fears. Everything else is learned. Your body programmed itself to be scared of certain things based off of an experience that you had at some point in your life with that, relating to different fears. So with this stuff, just know what just know these things. Know the definitions. What are they responsible for? You don't have to go much crazier into that. So fear and anxiety, stress response initiated when a situation perceived as threatening. The amygdala is the structure responsible for the fear response. The higher cortical areas integrate the sensory info, so what are we getting from our environment, with the learned experiences. So what happened to you before when you encountered a similar situation, and then it produces it to the amygdala. Amygdala stimulates the hypothalamus to activate your fight or flight by your sympathetic nervous system, kicking in your anxiety attack. Hippocampus provides negative feedback to try and attenuate that stress response. Hippocampus is going to your frontal cortex. Okay, I know you can talk yourself down and you can do things. It's like when you have bad thoughts, yes, you can push an old person down the stairs when you walk by, but your frontal cortex will be like, no, don't do that. You see the difference? Like, your frontal cortex will still limit you from doing things, which is why young people don't have a developed frontal cortex yet, and you gotta do stupid stuff. Yes?
Is that the same reasoning for like when you hold a baby and you can be like, I can drop this baby right now?
Well, yeah, it's called call to the void. It's like a weird thing. Yeah, you can. It's weird, but your brain thinks a weird thing sometimes, and your frontal cortex is like, no, don't do that. Aggression is linked to fear, which is why when people get scared, some people get very aggressive. Animals, when they get scared, they get aggressive. They're all in that same location. Same thing with like drinking and alcohol and with like aggression. They're all in the same located parts of the brain, and they cause the same issues. Last little bit is on language and speech. How do we have the ability to comprehend language so you understand what I'm saying? And then how do you know how to respond to me appropriately? Okay, speech and language are mainly dominated by our left hemisphere. And we have 2 major areas responsible for language. We have Broca's area, which controls the mouth. Okay, so it tells you, like I'm speaking to you, my Broca's area is the one that's causing the motor so that my mouth, my lips, and my tongue can articulate what I'm talking about so you can hear me. That's the Broca's area. And Broca's area is up here in that frontal lobe. It makes sense it's in the frontal lobe because this is where the primary motor cortex is. The primary motor cortex controls all the parts of your body. And remember I showed you the other day the homunculus. We have the brain mapped out. We know what parts of the cortex do what. So Broca's area that controls the mouth is going to be in the frontal lobe where the motor cortex is. We also have Wernicke's area, which is this little orange area. That is on the sensory side because you're hearing things. So Wernicke's area, do you understand what I'm saying to you right now? Broca's area allows you to speak it. So if I ask you a question, and this is why when you guys get to history taking, you don't want to give closed-ended questions like, what day is today? Monday. What day is tomorrow? Tuesday. Those are closed-ended questions. Open-ended questions like, how do you feel today? Then you want your patient to talk more. Because if they have Broca's aphasia, they're not going to be able to speak anything. They're going to have like tip of the tongue problem. Like they know what the word is, but they can't get it out. So they'll give like one word every few words that are supposed to be there. Like you can say, what's the weather like outside? They'll say warm, humid, sweaty. They can't say any other words because of the Broca's aphasia. Wernicke's aphasia, if you have a problem there, I'll say, what day is it? And they'll respond, oh, you know, it's great outside, and I went all weekend, I did this with my family, blah, blah, blah. But that's nothing what you asked them for. Why? Because the comprehension area, the Wernicke's area, is damaged. They don't comprehend what I am saying to you. So, here's an example. You have your exam next Monday. When you read the questions, let's say your eyes are right. Here's your eye. You're reading the question. We know it's going to go all the way to your primary visual cortex, right? Then it has to come back forward so that you can speak if it was like a oral question. See how it works? It has to go all the way to the back of the brain and then come forward so that you can speak and articulate and respond. That is how we have speech and language. Broca's in the frontal lobe, and Wernicke's area is in the temporal lobe, near that auditory complex. It makes sense because that's where, like auditory cortex, that's where you're hearing things. It makes sense your temporal lobe is where you would comprehend speech. I mentioned that already. And we're done. So Thursday, quick lecture on immunology, then a review, and then Monday's the exam. Email me if you have any questions. I'll see you guys later.